all. This is Dr. Mobeen Sayed from Drbeen.com. Welcome to one more show. So while we were talking about all those uridines or methyl pseudouridine and their effect on the ribosomal slippage, here comes another mRNA vaccine that is approved now in Japan. It is created by a company here in California, and then with another company in Australia's partnership, they have launched it. And Japan has given the world's first authorization or approval for a self-amplifying mRNA vaccine. In simpler terms, before we look at it, a vaccine, mRNA, that has its own little printing press machine. And when that mRNA gets into the cell, that printing press becomes active and it makes more copies of the messenger RNA. And that is how the vaccine actually increases inside the cell. So let's look at this one. We were looking at various issues with pseudo methyl uridine and or methyl pseudouridine, and here we have another interesting vaccine. So these are gifts for humanity. They're continuing. This is an RDRP. It will become a little clear later on that why did I make it look like a hand. And quick references. So this is drbeen.com. In the description of this video, there's a link that gives you access to drbeen.com at a very inexpensive price. This is the PR news about this. Japan's Ministry of Health, Labor and Welfare approves CSL and Arcturus Therapeutics ARCT-154, the first self-amplifying messenger RNA vaccine approved for COVID-19 in adults. This is in Nature. Self-copying RNA vaccine wins first full approval. What's next? And it is a nice one as well. We'll look into that a little more. Then here is the preprint. This vaccine was trialed in Taiwan. So this is that trial here. I'm not going to get into a lot of the trial itself. I want to explain the mechanism today. The overall efficacy was about 56%, and the efficacy against the severe cases was, I believe, more than 90%. They said that the efficacy looks low because they had decided that even one symptom of COVID will be called COVID. And they said because of that, efficacy is low. But anyways, that's go through this, and there is a lot more data here. Then this is the Arcturus. Arcturus announces self-amplifying COVID-19 mRNA vaccine candidate. ARCT-154 meets primary efficacy endpoint in phase 3 study. 95% efficacy overall for prevention of severe COVID, 55% efficacy overall for prevention of symptomatic COVID. Study conducted when Delta and Omicron variants were dominant in Vietnam. Incidence of unsolicited adverse events with ARCT-154 similar to placebo. No reported cases of myocarditis or pericarditis. ARCT-154 to advance into a pivotal booster trial in a major market. So this is interesting that they're saying because it is self-amplifying, because it converts that cell into a printing press to make more vaccine, the dose that is given is tinier. And because of that, the side effects are less. Their side effects were similar to placebo. Placebo was saline in this case. We have to dig a little more deeper. I think we all are aware of how these vaccines work and what could be good and bad about them. So, but this is the news. Then here is a Wikipedia article, ARCT-154. This is RNA-dependent RNA polymerases. That is our hero of the discussion today. So if you would like to know a little more, and while we are here, let's read this quick blue line here. The structure of RDRP resembles that of a cupped right hand and consists of fingers, palms, and thumb subdomains. Then Arcturus is RNA and mRNA proprietary technology. So they have their own videos as well for how does this vaccine work. Please feel free to check that out. It is interesting. Then self-amplifying RNA vaccines for infectious diseases. This is a different article where they discuss various technologies for how self-amplification vaccines are made. So if you are interested. And then finally, as we would discuss in our study, in our talk today, the virus that they used for, or the RNA they borrowed from a virus, that is Venezuelan equine encephalitis virus. So with these references, let's start. So creating a self-amplifying mRNA vaccine, how did they make it? So they took an RNA virus. They took a virus whose own genome is in the form of positive sense RNA, because that is what is most closest match to the mRNA for the spike protein. So they took this Venezuelan equine encephalitis virus. This virus is one of the major encephalitis virus. There are, I believe, Eastern, Western, and Venezuelan equine viruses. They can cause severe disease in humans. But as you would see that all the viral genome is actually removed from the RNA, and what is left is further modified for the spike protein. So what did they do? They took the virus and they took the genetic material of the virus. Now the virus's genetic material, this specific virus, that has two main structures. So if you see here, this RNA of the virus, this is the starting end, this is the ending end, that is fine. Some untranslated regions, that is also not important for us. What is important is that it has an initial part, the genetic material, that makes non-structural proteins, enzymes of the virus. And then it has a structural part which gives the structure to the virus, the coat of the virus and the spikes and all that. In the non-structural part, there is an enzyme which is the RDRP enzyme. So the non-structural parts are actually four: NSP1, 2, 3, and the 4. 4 is the one that is RDRP enzyme. So if you are a scientist and you're making this vaccine, of course, what you'll say is, I'm going to use this RNA because it is already structured and it has this enzyme in it, which makes copies of this RNA. Right? So you already have a printing press attached with this. Why do you have it? Because the viruses want to make copies of themselves. And they won't just make the coat of the virus. They have to insert their genetic material in there as well to make a successful daughter. Because of that, they have genetic material copying machinery present within their genetic material. So then what happens? Imagine that virus arrives in a cell, any cell, the cell which is a healthy cell. When that virus would arrive in a healthy cell, the virus would give its RNA in the cell, and then our ribosomes, or the ribosomes present in the cell, will pick up this RNA and would start translating it. Translating means they would start making proteins that are encoded on this RNA. Of course, when the ribosome will reach NSP4, then it will make this RDRP. RDRP, as I said before, this is that hand structure-like molecule that has fingers, palm, thumb, subdomains. RDRP's function, this is interesting. RDRP's function is to then pick up this RNA, viral RNA, and then start making copies of that. So inside the cell, there are two factories that are going on once this happens. One factory is that our ribosomes are working with this RNA and making more and more proteins in the viral structures. The other factory is that virus has brought in RDRP. And when again, this RDRP is not with the virus, but it is in the genetic material of the virus. The first translation means making the protein. First translation has to occur. When that first translation occurs by our ribosome, that makes the RDRP. Once the RDRP comes into being, that becomes another factory. And what does that do? So here, if you see, this is an RDRP now. It will pick up the genetic material of the virus, and it would start making positive and negative sense. In general, it would start making copies of those genetic materials. Then what would happen is when the virus is built, it has a coat and the spikes and everything. Then this genetic material copies are now available as well, which will be inserted back into the newly formed daughters. A brain will be given to them, and then these are released in the wild to go and infect other cells. So this is a normal behavior. Now, what did they do? They said, let's take this virus RNA and let's remove all the structural proteins, the virus coat, the virus spike, all of those. Also, let's remove any replication related enzyme except RDRP. And insert the messenger RNA for the spike protein where the viral proteins were, or viral structural protein area was. So essentially, they have gutted the viral structure making proteins and replaced them with the spike making protein, but kept the virus's copy making system. The printing press is left. RNA dependent RNA polymerase. So once they make this structure, this RNA, they then put that in the lipid nanoparticles. There are actually two kinds of self-amplifying RNA protein vaccines or therapies. One is where the RNA is put back in the virus, and then virus becomes the vector to bring the RNA inside the cell. So virus acts as a lipid nanoparticle, if you will. And the other therapy is where they actually take this RNA, amplify it, make many copies of it, and they put them in the lipid nanoparticle. I believe that this specific vaccine that we're talking about, in this, they make these RNAs and they put them in the lipid nanoparticles. Okay, so once that is done, then what happens? Imagine now that this vaccine is given. When this vaccine is given, here is that lipid nanoparticle that enters the cell. When it enters the cell and it is sitting there, we've done this discussions many times in the past. This is called an endosome. And the endosome stays in there, slowly becomes acidified and then breaks up. When this endosome breaks up, the lipid nanoparticle breaks up, the viral copies are the genetic material is liberated or freed into the cytoplasm of the cell. Now, the same thing that I discussed before, the ribosome, our ribosome will connect with this vaccine RNA, which has this red part is the RNA piece. Let me zoom in a little. So this red part is the RDRP making enzyme, and the remaining is the spike protein. So when the translation occurs, the red part, when that gets translated by a ribosome, that makes the RDRP. So now there are two factories. Our ribosomes are making more and more proteins for the virus, plus virus's own protein RDRP is making more and more of the genetic material. In this case, it is vaccine. So there are two factories. One factory is making spike proteins, and the other factory, RDRP, is making more vaccine. So the printing press for the vaccine is running inside the cell, making more and more vaccine genetic materials, and then other ribosomes are picking up these genetic materials and making spike proteins from that. And of course, then we all have been talking about this for 3 years. Immune responses occurs, immune training occurs, and so on. And we were just talking about the methyl pseudouridine as well, and the ribosomal slippage on that, and making garbage proteins as well. So this, I believe that this vaccine is also contains those methyl pseudouridines. So that means that ribosomal issue will be true for this as well. Okay, so a little more zoomed in. We are done basically with the mechanism. So a little more zoomed in. Imagine that this is the modified RNA. The NSP4 or the RDRP enzyme is present, then the spike protein enzyme is present. The first translation that happens by a ribosome, when they read and make proteins, they will make RDRP. This RDRP will then recognize the area of the spike. Now, this is an important thing, that it will not pick up this whole RNA and make copies of that, because that would just make more and more RDRP, and the cell would just get into a huge problem. So what this RDRP does is it would connect with the part of the mRNA where the spike protein messaging is and start making copies of that. So they have instructions over here that allow this RDRP to bind here. So once the RDRP binds there, it will then, this RDRP would then make copies of this. And so here we have the copies. For those of you who are medical trained or studying, I have just made it this simple, but ideally RDRP for a positive sense RNA will make a negative sense RNA. Then the RDRP would work with the negative sense and make the positive sense again. So it is a two-step process. And finally, what would happen is that these newly formed copies of the RNA genetic material for the spike protein, they would start binding to the ribosomes, and the ribosomes would start creating more and more and more and more spike proteins. So now two printing presses are working. As I said before, our ribosomes working with these genetic materials making spike proteins. So that is one printing press going on. And then the RDRP is working to continue to amplify and increase the RNA. So in one of their articles, they said that normal messenger RNA vaccine would have that RNA decay over time. So it will not persist there forever. Well, there are studies that show it persists, some show don't. But this is the narrative that normally the RNA present inside the cell would start decaying and going away, and they don't want it to go away that fast, even after a small dose. So they are making this printing press that would just keep making more and more vaccine available or RNA available. So this is the discussion. Just very quickly, some things to look at. This is their vaccine preprint. So if you see in here, one technology being applied in this development is the use of self-amplifying mRNA that allows host cells to make copies of the vaccine mRNA, increasing the amount of protein produced with lower dose of administration of mRNA. So this is one. Then if you look at the results of this study, these are the folks who were participant in various phases, 154 vaccine phase 1, 25 phase 1 placebo, then phase 2, 227 and 75, phase 3A, 448 and 152, phase 3B, 8059 and 8048. One interesting thing is that individuals between 18 to, I believe, 50 had 100% efficacy. So then here is the safety categories and the other material. I would let you look at that. It would become a longer discussion, but I wanted to make sure that we have an understanding of what this mechanism is. So if I summarize it, that what this mechanism is, imagine the standard mRNA vaccine, conventional one that we have, Pfizer and Moderna, and imagine adding in a genetic material to create a printing press, which would make copies of that genetic material. So now when you give a smaller dose, you actually teach the cell, not teach, you equip the cell with a new printing press that would just keep making copies of the vaccine. What do you think? Is it interesting? Does it sound more scary than the other vaccines, or does it sound better? What is interesting for me in here, other than the technology, is their side effects profile is equal to placebo, and their vaccine efficacy for severe cases is as good as the other vaccines. So that was interesting for me. Okay, so with this, thank you very much. Please like, subscribe, and share. If you would like to support this work, there are links in the description as well. And the best is to get Dr. Bean's access. And I would see you next week. Have a nice weekend.